Chapter 7, Section 9, Stringent and General Stress Responses. Now you have to remember, under normal, quote unquote, normal conditions, bacteria are going to be happy. They're in an environment that has excess sugars, excess amino acids, polypeptides, the osmotic stresses, due to, solvent concentrations are such that the bacteria are happy, okay? Um, but then things change. Things are always changing. And unlike us, when things turn bad for us, what do? We get up, we go elsewhere, or we go eat, or we drink, or we do something to change. Bacteria are at the mercy of their environment. So, what we have is their quote unquote responses to changes in their environment. Changes in the environment could be, depletion of the sugars, depletion of the amino acids, change in pH, things like that. Well, the bacterial cells are going to go from happy to being stressed, which is why we call these things that we're going to be talking about here stress responses or stress survival pathways. These pathways are going to be a series of signal cascades inside the cell and collection of genes that will be turned on, turned off in response to whatever stress event is now occurring. So let's talk about these stress responses. We're going to use E. coli as our example. E. coli being an enteric bacteria, it's going to be in your gastrointestinal tract, your small intestine to a little bit, mostly your large intestine. In that environment, these bacteria are growing where there's going to be an overabundance of all sorts of nutrients. Okay? We can't absorb all that we take in our diet, so we pass a lot of it out in our feces. So that's where they are normally. Well, what happens when they get passed out in the feces? They now go from an environment of overabundance to an environment of scarcity. Well, under the normal conditions, when they're in an environment of overabundance in our gastrointestinal system, there's really no need for the bacteria to make amino acids. It's swimming in polypeptides and amino acids and all sorts of stuff like that. So why make it when it can just absorb it, take it up from its environment? Well, that's when it's happy and overabundance. When it's now in a scarcity or a time of limitation, it now has to switch from taking in and making its own. Okay, that's a big difference. So as we've studied this in E. coli, we've come to find out that there is a collection, a set of immediate responses, and then a little bit later another set, and a little bit later there's another set of responses. Cell signals turned on, turned off, genes that were expressed or turned off, genes that were off are now turned on, and all sorts of things like that. That's what we're calling this stress response. The immediate ones we're going to see once the scarcity stress event, starts the signaling, is that, and we click this, RNA and tRNA synthesis stops. Okay, ribosomes and charged tRNA or new tRNAs, not charged tRNAs, new tRNAs. If your cellular is living in a land of plenty, yeah, you're going to be making as much protein as possible, as fast as possible, because remember, you're dividing every 15 minutes. So you're going to be making as many proteins, transcription, translation, as quickly as possible. But when you now are in a limited environment, scarcity environment, well, you don't need ribosomes, you don't need tRNA as much because you're going to have to slow down. because instead of just absorbing in all your amino acids, you're going to have to start making your own. So we see that protein and DNA synthesis will also stop. A lot of the metabolism, a lot of the energy is going to shift over to making new amino acids. Because remember, if they're not there for you to, absorb or to transport in, you got to make your own. Now, once the amino acid, the self-made amino acid levels start to creep up, so you now have pools that are creeping up, then we're going to see ribosomes, new ribosomes being made. rRNA synthesis, new ribosome production will also slowly start to creep up. Whereas before, when there was excess, it was make them as fast as possible. Make, because remember, every 15 minutes, boom, 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 boom. The signal for this switch to go from excess to limited or scarcity metabolism, you're going to see that these are what are referred to as Alloramones. I'm sorry, Alarmones, whatever that is. Basically special type of nucleotides, specifically guanosine tetraphosphate and guanosine pentaphosphate. So it's the normal G nucleotide, but instead of one phosphate, you're now going to have 4 or 5. So what we see happens is that under normal conditions, remember back in chapter 4 talking about the different growth curves. You have the log or the exponential growth, stationary, and things like that. Well, growth, and then scarcity. The stringent response kicks in, where a shift from making as much protein as possible, shift the metabolism from that to making as many amino acids as possible, so that at a later time point, you can get back into making more RNAs and proteins all over again. That tetra and 5-peptide, I forget what the tetra and the pentaphosphate, those levels are extremely low under normal happy conditions. When this stringency kicks in, that's when the tetra and the tetra and pentaphosphate nucleotide levels kick in, and it starts creeping up. These levels creeping up are going to shift metabolic functions away from RNA and protein synthesis and shift it towards amino acid synthesis. Once the amino acid levels get up high enough that more ribosomes can be made, things like that, we see that these levels start to go back down. Here's the tetra, PPGPP. We see here's our normal nucleotide. There's a hydroxyl there, so it's an RNA nucleotide. And there should only be one phosphate here up on carbon 5. But there's a second. And down here off of carbon 3, there's 2 there. There's a third one here if we're doing the pentaphosphate. What we find happens is that under normal conditions, when there's enough amino acids, ribosomes happily going along, the messenger RNA making new polypeptides. Okay, notice here, relay. Relay is a portion of the small subunit here. It's a protein, interacting with or complexed with the small subunit. Under those limited or scarcity conditions, what happens is that the tRNAs, here the J, up here the J with the AA, so it's a charged tRNA. Here it's the tRNA, codon, anticodon, just like we learned in genetics, cell biology, intro cell and molecular, but it's uncharged. It means there's not enough amino acids coming in from outside the cell to charge all available tRNAs. This will cause the ribosome to halt. No translocation will occur because there's no amino acid to transfer over to the polypeptide. No going from the A to the P site to the E, any of that. No translocation. The halting of the ribosome will cause REL-A to strip off phosphates from ATP and add it to GTP. So it takes off the terminal 2 and adds it, or the 3 and adds it. Increasing levels of the tetra or the pentaphosphates trigger all of these responses. Decrease in tRNA and tRNA synthase, ribosome, amino acid biosynthetic operons all become activated. So that means all the genes that are responsible for making the enzymes that will then, catalyze the production of amino acids, all of those operons will be triggered because the increased PPGPP or PPPGPP, the tetra or the pentaphosphate. Cell division's arrested, and all the other operons that go along with the stress response will kick in. Now, as I said, E. coli, it's one of the big stresses that we've studied has been, reduced nutrients, because it passes out with the feces. Another one is Caulobacter, which is another enteric. Carbon ammonia starvation. Okay, so no carbon source, no ammonia, no nitrogen source will trigger amino acid limitation responses. For Caulobacter, instead of just like E. coli, where you get this whole sets of genes turned on and signal pathways, metabolic pathways shut down, another one here for Caulobacter is it's going to increase motility. These bacteria are going to try to swim to or away from scarcity to abundance. It's a last-ditch effort to go from where there's nothing to a place where there's something. Okay, dropping out of the enterics. Let's talk Mycobacterium tuberculosis. Under normal conditions, Mycobacterium, once it starts growing in your lungs, starts causing the damage. We're going to see that the lungs are going to become hypoxic, meaning low oxygen levels in the tissues. There's going to be low oxygen levels even in the alveoli, the air gas spaces, because, there's so much fluid building up there that's going to be blocking bronchioles. Phosphate limited, nutrient limited, going to trigger responses where it goes from happy cells that are growing, granted they're growing slowly. Generation time for Mycobacterium tuberculosis is at 18 hours. And then it's going to switch over to what are referred to as persistent cells, where now it's going to be days between divisions. Side note, from a clinical standpoint, it's this damage to the lungs that we see here and the switch, the stress response induced switch from, the happy cells to this persister cells is also what gives them their resistance to antibiotics, because these persistent cells that are growing so slowly, turns out that, you take the pill today and within a few hours, your body's already turning it over, removing that antibiotic from your tissues, which is why you have to take antibiotics multiple times a day or take mega doses once a day. Well, they're so metabolically inactive that the antibiotics come and go before any metabolism is occurring. Figure from the textbook 725 is just showing you normal, limited, stringent responses. E. coli, Caulobacter. Sorry, I said Caulobacter is an enteric. It's not an enteric. I don't know why I said that. It's a gram-negative soil microbe. It's one of those that has the stalks. In stress response, the stalk releases and then it swims away somewhere else. And then will attach and live there happy for a while until stress response swims away, goes somewhere else. I misspoke. We see that growth curve that we talked about in chapter 4. Lag, exponential, stationary, decline. Well, it turns out that stationary, it will induce a type of stress response because the nutrients are now limited. Okay, there's not enough nutrients there to maintain that exponential or that logarithmic growth. So there's going to be a switching of what genes are there, what metabolic activities are active. RPOS is one of our proteins known as sigma factors. Sigma factors control regulons. Think back. Regulons. An operon is one set of genes that need to be expressed all at once due to one stimulus. Well, regulons, remember, could be multiple operons or partial operons, or operons that have been split apart and moved all over the chromosome. So sigma factors control regulons, okay? Individual genes, operons, whatnot. So RPOS regulon includes about 400 different genes. Genes that, will be expressed due to nutrient limitations. So you get this switch from, taking in amino acids to synthesizing amino acids. It's going to increase resistance to DNA damage. Biofilm formation, those genes responsible for that are going to kick in now. Osmotic pressure, oxidative damage, acid stress, all of these are going to be under control of this protein, this sigma factor known as RPOS.